Welcome to our second video of 8.2 cellular respiration, and in this video we are kicking off straight into the Krebs cycle. Okay, so in our previous video we looked at glycolysis and then the link reaction. So we ended up at acetyl-CoA. Now we are starting the Krebs cycle with our acetyl-CoA. So you'll see that the Krebs cycle, also known as the citric acid cycle, is a circular or circuit metabolic pathway rather than linear like glycolysis was. So this has got several names, the citric acid cycle being one of them, but it's most commonly called the Krebs cycle. It's called a cycle because it's cyclic. And we did look at those metabolic pathways in the past. Terrible slide here because all my text has come up as once. But you do not need to remember the names of all the compounds formed in the Krebs cycle. It's important to understand the overall process, and you must be able to recognize where a decarboxylation and an oxidation reaction has occurred. The Krebs cycle is a metabolic process that completes the breakdown of glucose. It occurs on the mitochondrial matrix, which we'll look at the mitochondria later, so we can actually look at the parts of the mitochondria. And it starts with our acetyl-CoA molecules that were produced as the link reaction. I've said all that already, just not in as technical terms. Alrighty, so let's have an overview of the Krebs cycle. Our acetyl-CoA enters the Krebs cycle and reacts with 4 carbon compounds to form a 6-carbon compound called a citric acid. Our CoA is released and recycled back into the link reaction. The citric acid then loses a carbon dioxide molecule, so that's that decarboxylation, and becomes a 5-carbon compound. This 5-carbon compound then loses another carbon dioxide to become a 4-carbon compound, as in the start of the cycle, so it's cyclic. At different stages in the cycle, compounds lose hydrogen, which comes out in the form of our electron carriers, that NADH plus H, and also FADH2. It's just another electron carrier there. Alrighty, so that was brief. The Krebs cycle was fairly brief. Now we've got oxidative phosphorylation, also known as the electron transport chain, or the electron train. So we've looked at glycolysis, we've looked at the link reaction, we've looked at the Krebs cycle, and at the end of the Krebs cycle we add reduced electron carriers that are coming into our electron transport system. So oxidative phosphorylation follows the Krebs cycle, so comes after the Krebs cycle. It is the process that is responsible for the production of the largest yield of ATP. It occurs in the mitochondria of the cell, and it is aerobic. This process requires oxygen. It occurs specifically in the mitochondrial cristae, and it is how electrons are passed along a series of membrane proteins. These membrane proteins provide energy for the pumping of protons into the intercellular membrane space so that the chemiosmosis and ultimately ATP synthesis can occur. Okay, so this electron transport chain, what do we mean? The electron transport chain involves the movement of electrons along the mitochondrial membrane. This facilitates the movement of protons, which eventually leads to the creation of a concentration gradient and thus the production of ATP. So the electron transport chain is a series of stages. So the first stage of the electron transport chain, reduced electron carriers from the link reaction and the Krebs cycle, so our NADH and our FADH2, are passed along the carriers. Okay, so these are located in the mitochondrial membrane. So the protein carriers that you can see in the diagram there. Our second step, as the electrons are passed along, they lose energy. They move from a high energy state to a low energy state because as energy is used up to pump protons from inside the matrix to the intermembrane space. Okay, so they use energy is used. It is an active process. Step 3, over time there is an accumulation of protons, and this leads to the development of a high concentration gradient of protons in the intermembrane space. Make sure you're looking at the diagram there to link what I'm saying. Once a high concentration is established, chemiosmosis occurs as the protons diffuse back into the matrix through the channel protein ATP synthase. Fifth step, as protons move through the ATP synthase, it rotates, which in turn causes ADP to be phosphorylated to ATP, that is because phosphate is added. Okay, so let's have a look at this process of chemiosmosis. You know I love the word osmosis. So, electrons are passed along carriers. Oxidative phosphorylation involves the movement of electrons along the inner mitochondrial membrane, which eventually generates ATP. Protons are pumped across the membrane. Electrons are released from reduced carriers and taken up by a series of pumps on the inner membrane. A concentration gradient is then established. As electrons move between the pumps, hydrogen ions are pumped into the intermembrane space. Chemiosmosis occurs. I can never pronounce it. Chemiosmosis is a diffusion of protons down their concentration gradient through the ATP synthesis generating ATP. Through ATP synthase generating ATP. Hydrogen ions accumulate on the inner membrane space. Hydrogen ions then move via chemiosmosis through the ATP synthase back to the matrix. The matrix. I'm thinking of the movie. So this is showing us chemiosmosis here. Sorry, let me go back to that one. This is showing you full oxidative phosphorylation, but it's showing you how the electron transport chain is the first part of that, and chemiosmosis is the second part, but the whole process is oxidative phosphorylation. Okay, so then the final steps. Once the electrons have reached the end of the electron transport chain, they are accepted by oxygen. This is why oxygen is referred to as the final electron acceptor, and this is why oxygen is needed for respiration. Oxygen acts as the final electron acceptor. When oxygen binds to the hydrogen ions, the protons, it removes the hydrogen from the matrix and so maintains the proton gradient for chemiosmosis and the production of ATP. So a summary of oxidative phosphorylation. Oxidative phosphorylation is the last process of aerobic respiration and occurs in the mitochondria of cells. It involves the movement of electrons along the inner mitochondrial membrane, which eventually generates ATP. Electrons are released from reduced carriers and are taken up by a series of pumps on the inner membrane, which pump the hydrogen ions across the membrane into the intermembrane space. As electrons move between the pumps in the cristae, their energy levels drop. Hydrogen ions accumulate in the intermembrane space. Hydrogen ions move via chemoosmosis through the ATP synthase back to the matrix. Chemoosmosis is the diffusion of protons down their concentration gradient through ATP synthase, generating ATP. Oxygen acts as the final electron receptor, forming water. Now, what happens if no oxygen is available? Well, the electron transport chain stops. NADH is not converted back to NAD+, and FADH is not converted back to FAD. If no NAD+ is available for oxidative decarboxylation, that link reaction, the Krebs cycle cannot occur. Glycolysis continues because oxidation is not required for glycolysis. Okay, so as I said, this is an overview of ATP production during aerobic respiration, and you can see each step of our respiration there and how much ATP is produced. Again, just a different pictorial view there, but it's also showing where these things occur, whether in the cytoplasm or in the mitochondria. Now, talking of the mitochondria, you need to be able to identify them and draw them. Here we've got a mitochondria. You can see the intermembrane space, the matrix, the cristae. You can see some ribosomes, some granules, some ATP synthase. Inner membrane, outer membrane, and some DNA in there. Remembering that our mitochondria does have its own DNA. That takes us back to the endosymbiotic theory. Here we've got an electron microscope picture of a mitochondria, and we've got a diagram of a mitochondria there. Alright, so structures of the mitochondria. We've got the outer membrane that encloses the mitochondria and keeps it separate from the cytoplasm, so the cytoplasm of the cell. We've got the cristae. So the inner mitochondria membrane is folded into these projections called cristae, and they're like fingers that stick out inside the matrix of the mitochondria. They increase the surface area of the internal mitochondria membrane, and that increases the rate of oxidative phosphorylation via the electron transport chains. The matrix is the fluid matrix inside the mitochondria, and it contains all the enzymes needed for the reaction of aerobic respiration, in particular for the Krebs cycle and that link reaction. And then our intermembrane space. The intermembrane space is very small and enables the build-up of a high concentration of protons needed for chemoosmosis. We also have mitochondrial DNA, that's often called mtDNA, and this is for protein synthesis. Hope you enjoyed those 2 videos. I'll see you in 8.3.